Should all get a message that that it's recording. Okay, then welcome everyone to the seminar this evening, or webinar. So, okay, someone else coming in. Okay, hi. Was just going to get started. So the incentive was basically just to show everyone that there's more to beneficials than bees, which most people might be familiar with, or everybody is nearly familiar with bees. And I found, especially in the last while, when I was very much confined to just the pretty surroundings of my house, that I got a bit more aware of what's crawling around outside, and maybe people who have a garden might have started growing vegetables or flowers. And there's a good few invertebrates which can provide beneficial services for you. So hopefully some of the animals which are going to be presented to you this evening, you might find in your gardens if you look for them and if you know how to look for them. So we will start the evening off with Aidan O'Hanlon, who's the curator at the Natural History Museum in Dublin, and he will talk about burying beetles. So I'm gonna mute myself as well now, Aidan. I guess you have a better chance of getting across. I'll hand that over to you.
Thanks, Inga. I'll just have to try and do share screens for a minute now. I can't. Only you can. So if you want to come back to me, and if you have the one I sent you that was recorded, maybe you could just play that one. It has narrations pre-recorded over it and stuff. You're still muted there, Inga.
Okay, Aidan, I think I've just allowed you to share it there. Try it again.
I got it now, yeah. I have the permissions.
Okay, good.
Grand. Give us a sec. Open share tray. Okay, does that look, can you see stuff from my screen? Is a presentation showing up? Oh, sorry, hold on, I'm muted. Is the presentation showing up now? The screen's sharing, or?
Yeah, it is, yeah.
Okay, cool. Grand. Well, thanks anyway for tuning in. Thanks, Inga, for asking me to do this. It's going to be an interesting series of talks, and I get to talk about some of my favorite animals, which are these guys, are carrion beetles. So I'm Aidan O'Hanlon. I work at the Natural History Museum, and I'll just jump in and get straight into the carrion beetles. So what they are is a taxonomic family, the Sylphidae. There's about 200 species worldwide, which isn't a lot when you think about beetles. Most of them, interestingly, are in the northern hemisphere. So unlike almost all other animal groups that have their species diversity in the tropics, for some reason the sylphids have their species richness at higher levels in the north. There's a hard break in the family between the subfamily, the Sylphinae. You can see one here, if you can see my mouse moving around. That's a typical kind of Sylphinae. They're called large carrion beetles. And then the other side of the family are the Necrophorinae, which we call burying beetles or sexton beetles in the UK. The Necrophorinae, I'll spend a minute just talking about their behavior and ecology because it's very cool. So they're well known for their habit of interring, embalming, and burying the carcass of a small animal, so something like a rodent or a small bird. They provide prolonged biparental care, which is very unusual in insects outside the Hymenoptera and unusual in animals just more generally. And they're ecological as nature's undertakers. So through their feeding and breeding activities, they free up nutrients that are locked up in an animal carcass and sort of recycle them back into the soil. So they're very, very important. This is what that looks like. This is basically what they do. So this cartoon shows on the left after
Aidan,
an animal has died.
Aidan?
Yeah.
The slideshow isn't showing, at least for me. I can just see the start page.
Oh, really? Oh, no. Is it the same for everyone? If there's a show of hands. Or...
That's it, yeah.
Yeah, sorry. It was only on the first slide.
Oh, I had gone so far. No, not really. I'd only got an extra slide in anyway.
It's caught up now for whatever reason.
Is it? It's, you know what it was? It was because it was in full screen. I'll just leave the if it's not too distracting, I'll just lead. I can't really leave them up. if I do this, does it show a picture of 3 mice?
No.
No, all right, that's very weird. I'll talk through it, and I can just move things around. Can you see me moving pictures around on this screen?
Yeah.
Okay, grand. So what was I saying? Yeah, so there's 2 major subfamilies, which are these guys on the right. These are the Sylphinae, they're called large carrion beetles. And then the other subfamily are the Necrophorinae, which we call burying beetles. And they might be familiar to some people. They're pretty distinctive looking animals with their orange and black bands. And it's these guys, the Necrophorinae, the burying beetles, that are known for their very cool and interesting behavior where they bury the carcass of a small animal and recycle its nutrients back into the soil. So this is way more messy now without animations, but I'll do my best. So on the top row here, this is what happens shortly after something like a mouse or a small bird dies. So a male and female carrion beetle will be attracted by the horrible smell of decomposing carcass. And they can fly in, they can sense these molecules from about 2 miles away, and they'll fly in, team up, greet each other, and get to work burying the carcass underground. But of course, in real life, it's not as simple as that. And what happens in reality is, because carrion is such a rare and kind of unpredictable resource, it's what ecologists call a bonanza resource. There's very fierce competition for access to the carcass. So the burying beetles will fight each other basically to win access to the carcass. So for some reason, males will only fight males, females will only fight females. And what happens is the larger males and larger females fight off the other burying beetles, and they get access to the carcass. So that's the first thing. But of course, it won't be only beetles that want access to a dead animal. Other insects require carrion as a food and breeding resource, like flies. Flies are great at getting into a dead animal. But the burying beetles have evolved this strange relationship with phoretic mites. So you can see in this picture, these are little hitchhiking mites that travel around on the back of the beetle. And as the beetle flies between carcasses, what they do is they disembark very politely, like they're getting off an airplane, and these mites, and they'll crawl around the carcass, and they're predatory on fly eggs and larvae. So it's a nice deal they've struck back in evolutionary time. So let's say they've done that, they've ousted the competitors, they start to dig the carcass and prepare it as a nursery for their offspring. And the way they can do this is by squeaking. So if we look at the bottom pictures now, male and female burying beetles squeak at each other constantly. So if you find one out in your garden or something like that, and you go to pick it up, it'll squeak at you. And it turns out there's a really nice study from about 5 years ago that looked at fossil specimens of carrion beetles from the Cretaceous, which is shown in this picture here. And what they found was that beetles from the Cretaceous had on their bum, basically, this is the first abdominal segment, they had these things called stridulatory files. So it's like a washboard structure in the right here, and on the left is this washboard structure in a modern burying beetle. And what happens is, as they wiggle their abdomen in and out, it scrapes off the bottom of their wing coverings and it makes a squeak sound. So it's called a stridulatory file, because it's the scientific word for squeaking is a stridulation. So they've been doing this squeaking behavior since at least the Cretaceous, which means that they were probably burying small bird-like dinosaurs, and maybe even our rodent ancestors. So the point I'm making is that they've been able to communicate this by squeaking for tens of millions of years. So they've been providing this ecologically vital ecosystem service for tens of millions of years. So they're squeaking, they're getting to work burying the carcass. The next step is they tear off the fur or feathers of the carcass and embalm it with antimicrobial secretions. So what these do is they stop a buildup of mold or bacteria, because they like decomposed carcasses, but they don't want them to decompose because they can't eat them. What happens then after that is the female goes off and excavates a little chamber outside this main area. It's called a brood crypt, and she lays her eggs in the extra chamber, and they hatch about 2 to 4 weeks later. And the larvae hatch, they're completely helpless, and what they do is they rear up their heads like this, and they perform a begging behavior. So the female or male beetle will then go and tear off chunks of rotten flesh off the carcass. I hope you've all already eaten, and then go and provision it directly into the mouth. So it looks gross when it's an insect doing it, but it's basically the exact same thing that birds do, that we think is cute. So like a chick in a nest, begging with an open beak, and then, you know, the mammy seagull regurgitates the fish into them. This is the same thing from beetles. So they're behaviorally very, very fascinating and ecologically really important. So I'll just end by talking a bit about what I've been able to learn about the Irish burying beetles, because for some reason no one had looked at them before. So I set to work reviewing them and gathering as much data as I could on them, basically. So we have 4 species of burying beetles. These are the Necrophoreinae, and it's worth pointing out that all 4 of these species will be engaged in that really cool sort of insect got behavior that I just spent the last few minutes talking about. They all appear to be pretty widespread across Ireland, if not under-recorded. There's no reason why they shouldn't be in every hectad really on the island, because they're quite geographically widespread. And they're active at different times of the year. I'll just highlight here these 2 species, Necrophorus humator and Necrophorus investigator. They're both about the same size, and they would use the same sized animal carcass. But much like what seems to happen in American burying beetles, they've evolved different periods. I mean, this is from what I could compile anyway. So Necrophorus humator and investigator a bit later, so they can avoid competing with each other. Yeah, next one. There's actually 5 burying beetle species. I'm still here, by the way. My computer's just telling me I was getting bad signal. If someone could give me a thumbs up.
Okay. There was just a bit of interruption, but that's okay.
Okay, grand, thanks. So there's an extra little bonus that we found. This is another new species called Necrophorus interruptus, which is new to Ireland as of this year. So I found when I was doing this work on the burying beetles, I looked through old museum store boxes and things like that, and I found 3 specimens of this guy. So it's very rare in Britain, and it's rare in continental Europe, and it looks like it was rare in Ireland. We only know it from these 3 specimens, one from Arklow, Killarney, and Clonbur and Galway. But it could still be out there, and just nobody's looking. So that's the burying beetle side of the family that do all the cool, interesting parental care and the burying and all that. We know a lot less about the other half of the family, and there are more species in this side of the family. These are the Sylphinae, the large carrion beetles, and they don't engage in that interest in burying behavior, and they don't have parental care, and they don't squeak at each other or any of that. And we know a lot less about their ecology and their behavior, but I've been able to learn a bit about their distribution in Ireland anyway, so it's a bit of a mixed bag. Some species seem to be fairly widespread, others have kind of unusual distributions, like this one on the bottom right, Eclipia opaca. It seems to be more or less associated with coasts and sandy soils. Maybe, we're not too sure. Then there are other ones like this, Dendrozina quadrimaculata. It's very distinctive. It's got these 4 spots, and it seems to be mostly associated with the south and east. And interestingly, this one only lives in really high quality deciduous woodlands. Another weird one is this Sylpha tristis that doesn't show up too well here, but it seems to be more northerly distributed. There's very few records, but that's how it is in Britain as well. It's only up in northern England and Scotland. And then you've got things like this, Phosfuga atrata subrotundata. I wish it had a common name. It comes in these black and brown forms, and it's very widespread across Ireland, and it's definitely our most common carrion beetle species. And here it is here, so I'll spend a second talking about it. It's our most widespread one, and it's probably uniquely an Irish species. So this was thought for a long time to be a subspecies of the parent species called Phosfuga atrata that lives in continental Europe and in Britain, and it's their commonest beetle there. But it doesn't live in Ireland. And this one, that's the Irish subspecies, Phosfuga atrata subrotundata, only lives in Ireland and doesn't live in Britain or continental Europe. And it was described as a species in its own right back in 1815. But that fell out of favor for some reason, and I think it's just an issue of semantics. There was some confusion over the fact that it's got black and brown forms, and it's just one of these things that's lost to history, but I'm quite convinced that it is a uniquely Irish beetle species, or has its origins in Ireland, but we won't know for sure until we do some DNA work. And to make things weirder, it's not even really a carrion beetle. It is taxonomically, but it feeds on snails. It's an active predator of snails and slugs. So it has this long head to reach inside snail shells. And here's a picture just of one of the specimens from the museum that was preserved like this. It's glued to a piece of card, and they preserved it with its head reaching inside a snail shell that I thought was pretty cool. So are there any conservation issues? Just to finish with the burying beetles, we don't know in Ireland, but we can look to America and look at populations of some of their species. So this guy here on the left is a species called Necrophorus americanus, the American burying beetle, and that's me holding one. They're very large insects, and it's critically endangered, and it's thought that its decline was associated with the extinction of this fella, the passenger pigeon, which went extinct back in the early 1900s. But it's now kind of agreed that it's near extinction here. The critical endangerment of this species is just down to habitat fragmentation, and we don't know much about that and the impacts of that on species in Ireland, but we do know that habitat fragmentation is terrible for biodiversity generally. And here's a screen grab of just a random section of the Midlands. It happens to be County Offaly. Nothing against Offaly. It was just a really nice picture showing kind of a general rural landscape and how badly fragmented it is. So we don't know if that's bad for burying beetles, but it probably is bad for all insects. And that's it in summary. The conservation issues, we're not too sure. If I had to guess from what I've put together, all these species on kind of the left-hand side of the chart here are probably doing pretty well insofar as they're geographically really widespread, and we've got contemporary records for all of them. These species on the right in red for danger, I'm a bit more concerned about. So this one that I mentioned that lives in deciduous forests, with two exceptions, two single specimens from Northern Ireland in the past 40 years. It hasn't been seen in Ireland in 85 years. The same with this one, Eclipia opaca, that lives around the coasts, with a single exception from Northern Ireland in 1970 or something. It hasn't been seen in 80 years. This one, Tyrolensis, that has the kind of scanty northern distribution, hasn't been seen in 117 years. And ironically, the one that's I found as a new species hasn't actually been seen in at least 92 years. But then just in yellow to show off, this is a species, Silpha tristis, that if you were going off just the numbers and the distribution maps, you might say doesn't look very good. But that I know for a fact just has a kind of a patchy distribution, and I found these thriving out on the Bull Island in Dublin just this summer. So maybe some of these have a patchy distribution, but again, they haven't been seen in so long that I don't know. It doesn't look great. So that's the state of knowledge. We've got 13 species. From these maps are put together from about 1,000 records, covers about a third of Ireland's terrestrial area. And we've got burying beetle and carrion beetle records now from every county and vice county except Longford. So if you're tuning in from Longford or know people from Longford, do keep an eye out. We were supposed to have a workshop. I was going to run one with the National Biodiversity Data Centre, but it obviously got put on hold because of COVID. And the idea was to get a whole bunch of contemporary records to really be able to answer what are there conservation issues, and can we get records for some of those species that we haven't seen in a long time. So that's it. You now know as much as anyone in the world does about Irish carrion beetles. So keep an eye out, and if you find any, or if you have any questions about them, that's how you can get at me. And thanks to Inga for organising and for you listening. This will all be out as a review. So if you really think burying beetles are cool, stay tuned for that, or you can ask me and I'll send you a copy. And that's it. Thanks. I'll stop sharing my screens now.
Okay, thank you, Aidan. If anyone has any questions for Aidan, please feel free to either click on the raise your hand symbol and then you can unmute yourself and ask a question, or you can just type it directly into the chat box, or just email Aidan if you have a more personal question or whatever. If not, I can move on to the next speaker, which is Peter Lillis, who is doing a PhD in Maynooth University. Okay, so I guess there's no questions at the moment. Peter, do you want to quickly into... Oh, Conor, you have a question. Okay, go ahead.
Hi. Yeah, sorry. Actually, what would be the kind of indication signs of if you for them like having to have buried a carrion? Like, is there any kind of like indication that they've entered the soil, or is there any kind of discoloration? You can watch over like what's kind of, if you're in the garden, what are you looking for in general if they're there?
I don't think you would know. I think they're so good at it that it's gone. They can bury it in about half a day and it's gone completely. And they move them as well. So if something died on grass or on a rock, we don't know how they know how to move it or why they should move it, but they will move it to somewhere that's more suitable to bury. So an area of loose soil. So you wouldn't find it. Yeah, you wouldn't find what they've. Well, maybe by the smell. If you went digging, if you smelled something awful and went digging around, you might find it. But
cool. Thanks.
Okay. Any other questions? Okay, Peter, do you? Karzan. Do you want to click on mute there, Karzan? Should be a little microphone symbol there in the top half of your screen in the top right hand corner.
Can you hear me now?
Yes.
So I'm just interested in sampling, Aidan, so what would be the ideal method for collecting these carrion beetles? I know like you have been using like pitfall traps for a while, but are they collected in any other methods?
Yeah, I used to study these when I studied up in Belfast years ago, and what we would do is get like a coffee cup or a plastic coffee cup, hang it from a tree with a funnel in it, and put in a piece of chicken breast. So you leave in the chicken breast, like fresh chicken breast, for about 2 weeks, and it gets really, really liquidy and smelly and awful. And you leave it up for 2 weeks, and then go and collect it 2 weeks later, and it'll be crawling in them. And flies and other things as well. So definitely some kind of baited trap, like using some meat, like gone out rotten meat. So it doesn't smell nice, but unless you're a burying beetle, then they think it smells lovely.
Okay, thanks, Emilia.
Okay. Peter, do you want to say a few words and I load up your presentation there?
Yeah, sure. So thanks very much for having me. I am currently doing my PhD down in Maynooth University, and I work on entomopathogenic nematodes. So they are obligated parasites of insects, and they're really effective, and they're found on basically every continent on Earth except for Antarctica. And yeah, they're very effective at what they do, and there's a lot of parallels with what Aidan was talking about finding a dead or finding a host, and there being a lot of competition. But yeah, they're really common, and they're very common in Ireland, but I don't think most people know about them, if they're aware at all about entomopathogenic nematodes. So yeah, I just kind of tried to do an overview and then specialize in on some of the nematodes that we actually find in Ireland here through previous ecological studies.
Okay, thank you. Is the screen sharing on at the moment? Can people see the background?
Yeah, the screen sharing's on, yeah.
Okay, I hope the sound is on as well. If not, let me know.
No. No, there's no sound there.
No sound, okay. Just a second.
Which is honestly fine, because I hate my own voice, so I can talk over it and try and match the video if you want.
Include computer sound. Okay, that looks better now. Let's try it again.
And you're just not screen sharing there?
There's no screen share now. Okay, wait. Sorry about this now. I've practiced it and somehow it still isn't working. Okay, is it there now?
Yeah,
okay.
There now.
Down at Maynooth University, and I'm based in the Behavioral Ecology and Biocontrol Lab. So the basis of our lab is...
I can't see video there, sorry.
Pathogenic nematodes.
Yeah.
Video's gone again.
Yeah, video's gone, yeah. That's good.
Really promising alternatives to chemical pesticides.
Yeah.
They are small roundworms which live in the soil, and they seek out insects to infect and kill. The reason that I'm interested in them is because I am fascinated by parasites and the way that they are able to exhibit really complex behaviors with a very simple body structure. So this is Galleria melanella. So it is an insect that we use in order to culture up these parasites. And we might expose this insect to about 100 IJs, or infective juveniles. And after a little while, and if you're squeamish, you might not want to look at this, the insect will basically burst open and reveal about hundreds of thousands to millions of new IJs that are ready to infect new insects. So you can see that these IJs give really good protective properties to the soil because there's an alternation of these generations. So successive generations in the field can get rid of pests for quite a while. So you might be familiar with C. elegans, so it's a really common model used in science. So they start off as an egg and they go through different molts or stages of their larvae, and they go from L1 to L4 and they become an adult, which is sexually mature, and they will produce new eggs. But in C. elegans case, which is a free-living nematode, it's not a parasite, they can undergo what's known as the dower stage, and this dower stage is an abiotic stress-resistant stage of their life cycle. And they can adopt this, and it's non-feeding, so it needs to find a new source of food in order to become an adult. And this is very similar to the entomopathogenic nematode life cycle. So they start off as an egg and they molt through successive generations or stages in order to become an adult. But once the nutrients of the insect host is used up completely, then the IJs will begin to develop. And this IJ is similarly an abiotic stress-resistant stage, and they will burst out of the insect and find a new host in order to infect. So you can see here, this is a Galleria melanella that's infected, and it's got a pink color, and I'll explain that in a minute. And these nematodes will multiply until all the nutrients are depleted, and they leave the host. And even if just a few of them are able to find a new host, this cycle can continue again and again. So they're really effective. These EPN associate with a symbiont, so the guts or a vesicle has a bacteria in it. So it's Xenorhabdis in the case of Steinernema, or Photorhabdis in the case of Heterorhabdis. But what that means is these IJs have bacteria inside them, and once they get inside an insect, they basically ingest this bacteria in order to kill the insect by sepsis. So the interesting thing about this bacteria is, quite unusually, it is bioluminescent. It glows really strongly. So you can see here a Galleria that is infected, and when it's put in the dark, you can see that it's glowing. And we're not quite sure why the bacteria cause the Galleria to turn a very dramatic color or glow in the dark. So there's a couple of hypotheses. It could be a signal to scavengers to not take a bite out of this insect. So this has been shown in toads, and in birds, they dislike the red color that H. megadus and H. bacteriophora turn the insects. It may attract other insects to infect, like a moth to a lamp. Or it may be a sink for free radicals that are produced as a result of metabolism. So why are we studying EPN? Well, they could be really good alternatives to chemical pesticides. But the other reasons are they are really good for studying free-living or plant parasitic nematodes, and most importantly, they could be used to study mammalian parasites. So mammalian parasites are quite hard to study because you need to infect mammals in order to study them. So if we're able to use the infective juveniles of these insect parasites instead, that would be really good. So I'd just like to talk about Bull Island if we're talking about the invertebrates of Ireland. Bull Island is an island off the coast of Dublin, and it has a lot of different animals on it, as you can see here from this Dublin City Council poster. But in my opinion, I think they neglected to include one of the most beautiful animals on the island and one of the more interesting ones, which is the nematode. So there have been a number of ecological studies on the island, and they have found two different species of nematode living there, Steinernema feltiae, and quite interestingly, they found Heterorhabditis downsii. And the interesting thing about that is that, as I said earlier, these EPN IJs generally associate with one bacteria, which colonizes their gut or in a vesicle. But the H. downsii that were found on Bull Island had two different bacterial symbionts. There was Photorhabdis cinerea, which is the purple, and Temperata, which is the yellow. So it seems that the nematodes on the island had two different species of bacteria, and these bacteria could confer different advantages and resistances to these nematodes. So in other words, they are able to extend their ecological niche by associating with these two different nematodes. And this was shown just recently that the cinerea bacteria, the purple, has heightened antimicrobial and antifungal activity. So in other words, as the IJs get into the host, the bacteria will colonize the insect, but they need to make sure that no other bacteria can piggyback and start eating away at the insect as well. So they produce antibiotics, and the same for fungi. They can't let anything start to decay or start eating the insect as well. So they produce antifungals. And Temperata produce them as well, but not to the same extent. Whereas down at the bottom here, you can see that the Temperata have an increased light intensity at 48 and 96 hours. So whatever reason that these cadavers are glowing, they seem to do this more effectively. They also are more protective against co-infection with Steinernema feltiae, the other nematode that was found on the island. So quite interestingly, these two species of bacteria can confer different advantages to the nematodes, which is why they coexist. The focus of my research is to use omics technologies in order to study the molecular determinants of many of the behaviors that these nematodes exhibit. So I'll just give one example briefly. So we studied infective juveniles that were stored in water, and then we infected Galleria melanella with these IJs. And over time the Galleria became infected, and then we mashed up the cadavers. So we produced cadaver macerate, and we took the water from the IJs that were stored, and we dropped both onto IJs on a petri dish with no other stimuli. And what we found is, compared to controls, it caused a lot of dispersal in these IJs. So it seems that the IJs are able to respond to high levels of other IJs. In other words, whenever it gets very crowded, they're able to tell each other that we should leave, which might heighten their chances of finding a host and reducing competition. So we looked at the water that these IJs were stored in, and we found a large number of ascaricides. These ascaricides are sugary fat chemicals, and they have been previously shown to be signaling molecules. And what we were showing there is that the high levels of these ascaricides are correlated with this dispersal. So these may be implicated, and this has been shown since. So that was just a brief foray into the EPN world, and I hope that if anybody didn't know about entomopathogenic nematodes and indeed the war that's going on underneath our feet every day, I hope that now that was enlightening and not too disgusting. And I don't want these to be on posters for Dublin City Council, but yeah, I hope that was interesting. And I'd just like to acknowledge my supervisors, Professor Christine Griffin and Dr. Jim Carlin, and also acknowledge that my funding was from the John Hume Doctoral Scholarship in Maynooth University. So I should be about for any questions, and thank you very much for listening.
Am I still sharing the screen? I guess I am. Let me just see how I can get rid of that. Is it gone now?
Yeah.
Okay, great. So I have a question for you. If somebody else has a question, are those entomopathogenic nematodes available for purchase for people to put out in their gardens, similar to like Nemoslug or something, or?
Yeah, exactly. So Nemoslug is Phasmarabditis hermaphroditica, and that's really effective against slugs. But you can buy many different entomopathogenic nematodes, such as S. feltiae, S. carpocapsae, or H. megadus or H. bacteriophora, and they come as a powder. So I'm sorry in advance, I'm going to call them interesting and cool about 100 times when I talk about them, but one of the interesting things about them is that they can be dried down, so they can become basically a powder, and then whenever they're put into water, they will rehydrate. And they've actually found samples of nematodes that have lasted 39,000 years in this cryptobiotic state. So they're able to survive really desiccative or drought conditions by drying out completely. So yeah, you can buy them online, and you just fire them into a watering can and just put them over your flowers, and it'll give you really good protection against beetles. And the promising thing about these as an alternative to chemical pesticides is that they stay in the soil. So especially cruisers, they're called, like H. megatus and H. bacteriophora. They go into the soil and they try and find insects. So they're incredibly effective against beetles, and they will be staying away from anything like hoverflies, bumblebees, or honeybees, so they won't be trying to attack our pollinators. So that's one of the exciting things about them. So yeah, they're incredibly commercially viable, and they're available.
Okay. And how specific are they for beetles? So if you would put them down for, let's say, a type of weevil or something, would they go for other families of beetles as well? Are they pretty discriminant?
Yeah. So it depends. So some of them, so Steinernema scapteriski, I might be mispronouncing that because I don't work with it, but it is quite specific towards mole crickets, and they can be used on golf courses where they want there to be a lot of biodiversity, but it'll kind of kill all these mole crickets that are causing problems. Whereas things like Steinernema carpcapsi or H. megatus that I work with, they are complete generalists. They will absolutely kill anything they encounter. So yeah, they're not great for biodiversity in the soil in that regard because they will kill any insect they find. But because they're relegated to the soil level, they're much better for our pollinators than pesticides that are chemical based.
Okay, great, thank you. Are there any other questions? Yeah, Conor.
Yeah, cool. I actually had a question about you mentioned the bioluminescence deterring insects. Are there any insects like predatory or otherwise that still will attack a larva that's being targeted by these nematodes, or is it kind of a general rule if the nematode has made itself a home in there that they typically can, because of the bioluminescence and whatever else, that they don't get targeted, or is that part of the life cycle? Like is there any kind of observations on that? In terms of like upper scale food chains and stuff like that.
Yeah, so it's a really interesting story. Again, I'm going to call it interesting, but there's a really cool story at the moment where it's very difficult to figure out because obviously behavioral stuff in the field never works out because if you leave things out, other scavengers will get to them and everything. But so there's a doctor called Sulchuk, and he has found that these nematodes will produce an odor, and the odor actually repels most insects. So they're looking into purifying this, and they're looking into using it as a general insect repellent. But the interesting thing is that the bioluminescence of these, the bacteria that the IJs give to the insect and it causes the cadaver to glow, it can, in some cases, attract certain scavengers. So it's almost as if it is calling in a next wave of insects so that when the IJs leave the host, they're able to infect loads of other insects. But we're currently doing some behavioral work in the lab, and we're finding that some insects, if they're in the light and they're not able to see the bioluminescence, they're indifferent. They will eat the cadaver if it's infected, but if it's in the dark and they're able to see the bioluminescence, they will actually move away from it. So it seems that it could be a signal to not eat it until it's ready to infect. But we still need to look at this in more detail.
Okay, Karzan, do you still have your hand up from the last time, or did you have another question?
Sorry, Inga, I don't have any questions. Sorry for
okay. Yeah,
okay.
For some reason the hand is still showing. I don't know if it.
You can click it again to make it go down.
Oh, okay. Okay. If there's no other questions for the moment, then I'll load up my own presentation. Just a second. I hope it'll work out easier.
And I'd just like to say that in the presentation there, the references at the bottom got cut out. But if anybody's interested in any of the things I was talking about, just feel free to email me, and I can talk about it later on if anybody has any questions.
I'm sorry. There's a big mess going on now. Okay. Let me just try to share the screen in a normal way. Okay, so screen should hopefully be showing and sound should be showing. Can you see my desktop background now?
Yep.
Okay. So then I'll put on my presentation, and I'm sure it's going to sound a bit awkward as well because it's pre-recorded. So hopefully it's still easily understandable. My name is Inga, and I am a postdoc in the Applied Ecology unit in NUI Galway. I'm here on a Marie Curie fellowship, and I'm studying the diet of ground beetle larvae. As part of the same fellowship, I was in Oregon State University for the previous 2 years before being here in Ireland, where I was looking at different ground beetle species in agroecosystems. So now I'm going to talk about ground beetles, or Carabidae. Ground beetles are one of the most species-rich animal families in the world, with about 40,000 species. Of these, 2,700 can be found in Europe and 211 in Ireland. Irish species range in size from just about 2 millimeters to nearly 30 millimeters, and most species are dark-colored. These are generally active during the night, while many of the more colorful and metallic-looking species, such as the tiger beetle Cicindela campestris, which you can see in the top left corner, are day-active. If you have a closer look at this beetle, you will also notice that his eyes are relatively large in comparison to the size of his head. This is typical for day-active species, and I guess depending on the size of your screen, you might also notice that, for example, Noteophilus bigotatus and Elaphros cupreus also have rather large eyes. These are two further day-active species. Identification to species level is often difficult and relies on features such as the presence or absence of small hairs, or the dissection and examination of the genitalia of the beetle. Ground beetles have a few characteristics by which they can be distinguished from other beetle families, though you generally would need to look at them rather closely. These are a thread-like antenna with 11 segments, 5 segmented tarsi, which are the final segments of their legs, which I have shown here in red. And then if you turn the beetle around, the shape and size of 2 parts of their hind legs are also characteristic. The first one are the coxae, which I attempted to demarcate in red, and these are roughly triangular plates which divide the first abdominal segment in half. Then the trochanters, which are shown in green here, are large and lobed and extend along the femora of the hind leg. If you just get a superficial look at the beetles, then usually the flattened body, the long legs, and the more or less parallel-sided wing cases, which are called elytra, are some features to look out for. Another group of beetles that can be commonly found on the soil surface in gardens are rove beetles. Here, for example, is a devil's coach horse, and these can be rather easily told apart from ground beetles by the length of their elytra, which leave several of their body segments uncovered. Most species of ground beetle either breed in the spring or in the fall. Females will deposit between 30 and 600 eggs into the soil or into a layer of plant residue on the soil surface. After hatching, the larvae is the life stage in which the beetle grows, and they usually undergo 3 distinct stages. Larvae are mostly subterranean, and only those of a few species, such as Nebria brevicollis, which is shown in this picture here, can be seen above ground. As you can see, the larva looks distinctly different from an adult beetle, and it's during the next stage, the pupa, that the transformation takes place. Pupation generally occurs in the soil, and the newly emerged beetle is still soft and very vulnerable, and usually does not leave the pupal cavity until somewhat hardened. The second picture from the right is that of a freshly emerged Nebria brevicollis beetle, which is still nearly white and completely soft. After a few weeks, when the species is fully hardened, it looks rather different. While most species have an annual life cycle, and will generally die after laying their eggs, some larger species may live for longer and produce more than 1 generation of offspring. Ground beetles can be found in a large variety of habitats, ranging from woodlands to beaches and peatland. As with most other animal groups, they are species which are associated with particular ecosystems and that can only tolerate small changes in habitat factors such as soil humidity or pH. But there are also species which can be found in a range of different habitats and can be quite numerous in rather disturbed settings such as urban areas and agricultural fields. As evident by their name, ground beetles mostly forage on the ground, but some species are able to climb plants and trees, and some species are also able to fly. But this is usually only used for dispersal. Most ground beetles are omnivorous and feed on a variety of soft-bodied invertebrates, living or dead, and plant materials. However, species have different preferences. The limiting factors of their diets are generally the size of their mandibles, which can be seen on the pictures on the right. The top picture shows the mouth parts of a typical omnivore. These are effective for capturing a range of different prey, and the bottom picture shows very stout and strong mandibles of a species that also feeds on plant seeds. However, some species are not able to overcome the defenses of their prey, even if their mouth parts would be large enough to capture this. Back in Oregon, I was doing some feeding experiments with small slugs and a rather large species of ground beetle from the genus Calosoma, which generally feeds on caterpillars. Some of the beetles tried to attack the slugs but were unable to cope with the mucus which stuck their mouth parts together, and they didn't go near them for 2 weeks despite not being offered any alternative food. Ground beetles are often referred to as opportunistic feeders and mostly locate their food by random search, feeding on whatever is abundant that can be overcome. But there are also some more specialized feeders that can detect chemical cues from their prey, which then actively get sought out. Here are two of the more specialist species in Ireland. On the left is Cycrus caraboides, which feeds on slugs and snails, and on the right is Larissa pelicornis, which feeds on small soil organisms called collembola. Cycrus caraboides has a long and narrow head that it can fit inside a snail shell, and it also attacks mollusks in a way that will seize mucus production. Larissa has a lot of small hairs, called setae, on the lower segments of its antennae that help them capture the small prey and funnel it towards their mouth. Unlike, for example, parasitoids that are hugely effective on a particular pest group, generalist predators such as ground beetles are somewhat effective on a large variety of pests. They will feed, for example, on aphids, moth larvae, maggots, and slugs, as well as weed seeds, and are particularly useful at the start of pest activity, when they can delay or even prevent the growth of pest populations from reaching a level that will cause too much damage. But because they are generalists, they are likely unable to quickly reduce an already large pest population. As a bonus, however, a good few species are readily found in gardens and agroecosystems already. Chances are, if you have a garden, you will likely have ground beetles as well. To make it more attractive for them, there are a few easy things that can be done, such as providing undisturbed areas for shelter, such as piles of leaves, stones, or logs. Other options are to plant areas with perennial plants and grasses, or even build a beetle bump. This is a small version of a beetle bank, which is used in agricultural settings, particularly in the UK. These are raised mounds of soil alongside of fields that are planted mainly with native bunch grasses and offer the beetles and other beneficials a permanent, undisturbed habitat with drier conditions in winter and more even temperatures year-round. Another very important thing is to avoid the use of pesticides. These are toxic for many beneficial insects and can be taken up from consuming poison prey. Even better is that these simple management options will not only promote ground beetles, but other beneficial invertebrates as well, such as rove beetles, spiders, or centipedes. Here are just two of the beneficial ground beetle species you might find in your garden when turning over logs or stones. The first one is Rusticus millenarius, which is a mostly nocturnal species that is generally active in Ireland between June and November. And the second one, which actually has a very nice metallic hue in the right light conditions, is Carabus nemoralis. It is also mostly nocturnal, and adults are generally active from early spring until the autumn. Both of these species feed on a range of pests, including slugs. A different way of finding out which ground beetles you might already have in your garden, and footage from another common species will be coming up now in a short video. The easiest way to survey for ground beetles is to set up a pitfall trap. A pitfall trap is basically just any container, such a plastic cup or any other material. We just put it through a hole in the soil. And the very important bit to observe is that the rim of the pitfall trap is flush with the soil surface, so that the beetles who are walking around are not turning around because they've hit an obstacle. Because we're in Ireland, and it's likely that there might be some rain overnight, I'm going to put on a little roof. Yes, it's just a piece of plastic with some nails in it. You obviously have to leave enough space that the beetle can still feed through. Okay, now let's see what we found. I brought a little box so I can put the contents of the pitfall inside. Oh, and it looks like we have a good few beetles. Let's take out those clumps of soil. Okay. Now, let's have a look what we got here. You see lots of small, black, fast-running beetles. And given the time of year that we have for the autumn, and the way that these beetles are moving so fast, harvestman, come out of there. These are Nebria brevicollis, which is a very common species that undergoes a summer hibernation and then gets active again in the fall to mate and lay their eggs. So you can see how fast they're moving. They have reddish legs and they're otherwise pretty much brown, and they're usually active during the night. My glove, hopefully you should be easily visible against the white background. They're also known as the European gazelle beetle. I guess it's because they're moving so fast. And they're usually very gregarious, so they occur in large numbers. Here are some links in literature if you want to find out more about ground beetles, and also a link to the Biodiversity Ireland website where you can record species sightings. And finally, I want to say thanks to my funders, the European Union, for providing me with the Marie Skłodowska-Curie Fellowship. And I want to give credit to some of the amazing pictures I've used in this presentation, which are mostly from Udo Schmidt from Deutschland, but also some other providers which I've accessed through the Wikimedia Commons page. If you have any questions, please feel free to email me to my email address below. Okay, so I'm glad that that's over. Let me just try to get rid of the shared screen again. Okay, is it gone now? Yeah?
Yeah. It is.
Yeah.
Okay, great.
Yes.
So, are there any questions? Karzan, is that a hand again? Anyone? Okay.
Yes, so just two questions, Inga, two quick questions actually. So are any of them like commercialized?
No. So as far as I know, in Ireland, there's no beetle as such for fit for purchase.
Yeah.
I guess a big difficulty with them is that they're very hard to rear in captivity. So the cost effectiveness of rearing an awful large amount of ground beetles would probably be forbidding, unfortunately.
Yeah, because one of the common beetles found with the Carabidi is rove beetles, and I know some of them, like one of the species called Athita coriaria, is commercialized. So I was wondering whether it might be possible for Carabidi as well, but probably they are generalists, that's why it might not be possible.
Yeah. No, as far as I know, there's nothing available.
Yeah. And you also, that was a nice video actually about collecting them in the field. Is there any particular or technique to differentiate them between, for example, some very similar beetles, in particular the immature stages? For example, sometimes you see a lot of like rove beetles in the field, in pitfall traps collected with them. Are they easy to be identified in the field, or do they need to be checked under the microscope in the lab?
Some of the larger and more common species, I guess, might be identified if you are kind of practiced. But there is an awful lot of different genera of beetles, for example, Amara species, which look pretty much identical unless you look with a really good microscope at a beetle that's actually holding still. I guess the problem as well is that they're moving so fast, so it's very difficult to actually get a really good look at them if you don't somehow fixate them.
Perfect. Yeah. Good.
Okay, if there's no other questions, then the next speaker is Conor Daly. Conor, do you want to say a few words beforehand, or?
Yeah, I can do. So this presentation is going to be about parasitoid wasps. So it's a group that's surprisingly diverse. So hopefully you guys might get an interest and want to find out more about them. I do have a reference list that I can share to anyone who wants to contact. So I have my email at the end of it, so you can contact me if you want the more research kind of information kind of things. But I hope you enjoy it.
Okay, thank you. Let's put on the shared background. And everybody able to see it?
No, not yet.
No.
Oh, it's not. Is it not visible?
No, not yet.
How is this so difficult? Share content. Include sound. Desktop. Can you see it now?
It's just loading. Yeah, perfect.
Okay.
Us here in Ireland, both from local level communities trying to get more diversity into their environment, and also economically, how they are vital for the future of agriculture in a lot of ways. So we're just going to give you a brief outline of my experience and where I'm coming from this, and then we'll get into it. So I studied zoology in NUIG, and I did a master's in biodiversity conservation in Trinity College, Dublin. That's actually where I did a bit of research into these guys. I looked into how they were affecting aphid populations in an organic oat field. So I've done science teaching at summer camps and secondary schools, and I'm currently doing falconry out here in the west of Ireland. And I am obsessed with these wasps. So I'm actually currently working towards getting funding for a PhD to research them. So what are parasitoid wasps? So if I say a wasp, you might immediately come to this image, this white and black beauty, big yellow, oftentimes aggressive, the instinct to duck or run away when you hear the buzzing, can get your blood pressure up and everything else. These are not what parasitoid wasps are. So the wasps that we are used to are the social kind. They live in nests and hives, and they work together as one big group, whereas parasitoid wasps are a lot more solo. So they basically look more like this. They're a lot more slender, and they have something called an ovipositor, this long extended appendage here, and it's basically for injecting an egg into another host. So that's what defines a parasitoid wasp. It's injecting an egg into a host, and the host is then dies when that egg emerges as an adult. So for a parasitoid, it means it's killing the host. Parasitic, as you might like any other kind of parasites, they don't kill the host. So a parasitic does not kill the host. A parasitoid does kill the host as a result of it being a host. Another thing about them that differs from normal wasps, like social wasps, is the body segments. There's very small differences. Some are small, some even so small you need a microscope to be able to figure out what you're looking at. Which makes it all kinds of fun to identify them. So as well as that, they are very, very tiny. The smallest on record has been less than 1 millimeter in length, almost 2.2 millimeters long, which is an insane amount of size when you think about it. But the bigger ones can be as long as 12 centimeters. So there's a huge range of size to deal with, and they are found throughout the globe. There's nowhere that's fully absent this species. So they are super weird, almost an absurd number of species, of body shapes, of lifestyle choices. As well as this, they also have HIV-like viruses that they use against the host to keep it from rejecting the wasp larva. So they are very, very unique. Dozens of different body shapes and also different ways of doing things. The bottom right there is actually a picture of a wasp emerging from a fully mummified aphid. So the insides have been completely cleared out. Wasp has had all his fill, and now he's emerging to go look for a mate and for injecting the eggs. So what do we know about them in terms of their biology? Quite a good bit, but a very kind of narrowed viewpoint. So they will occur wherever you find other insects. There's no insect that is fully independent from parasitoid intervention. There's always at least one wasp species that will target you if you happen to be an insect. So it's hard. So if you're in the bug world, you're not immune from this group of wasps. The ones we know the most about are your aphids and your caterpillars and some larvae. It's the insects that we have economic concerns about. So things like that do harm to crops and everything else. They're ones that we know the most about because they're an alternative to pesticides. They're arguably the most diverse group of insects on the planet. We're talking hundreds of thousands of species potentially out there. And as a result, they're an important indicator for the health of a habitat. If these guys can be found wherever other insects are, if they're not present, what's going on? What's happening in that habitat? So to give you a reference of how many species there are, for Ichneumonids, there's over 24,000 worldwide, Braconids 17,000, and Chalcids, there's only 100,000 identified to date, but there's estimates from 250 to 500,000 worldwide. In Ireland, obviously, the numbers won't be anywhere near that big, but still they would make up a significant number of overall insect population. If you took all the insects in the world and put them in a room together, in terms of species, over half would likely be parasitoid wasps. And these are just 3 groups. There's way more. This is just touching on a few of the main groups that we tend to know the most about, and books and stuff have been written on them in more detail because we know the most about them in a way, because they're some of the more diverse groups. So your ichneumonids more look like this. They're typically the larger wasps, so really big antennae, all different kinds of body shapes. So this is not a set body shape. There's plenty of different other appearances ichneumonids can have. The big thing that you can recognize is usually there's this little pteriole, this little circular thing made up of veins. It's usually with this one obviously has one missing here. It should have a little line here. That's telltale of an ichneummonid. That's more for identification more than what most people will need, but that's what you're looking at when you're seeing them. Then braconids, much smaller, very tricky to kind of determine what species you're looking at beyond simply that you think it's a braconid. There's a few physical similarities. Think of them like miniature ichneumonids is the best way to look at them. So as well with that, they've got the wings kind of flat over each other on their abdomen. That's kind of the only way to tell them that it is a braconid. And then your chalcids. These guys are even tinier. These are some of the smallest species you'll see. They just look like little flies. Like you'd easily mistake them for a fly on your hand, when in actual fact, if you have a closer look, you notice the wings and the body shape and the antennae, and all of a sudden you realize it's a little wasp. So in terms of diversity alone, the chalcids would be the most diverse group, and we're still studying how diverse they really are. So they're the kind of things you might be looking for, but that's kind of just to give an idea of what you might spot in your garden. So why do we want them in our garden? So they are a natural pest control. So if you're a organic cereal farmer, you have your oats and your wheats, you're going to have an aphid problem because pesticides would normally keep them at bay, but you're trying to avoid doing that. An alternative is wasps. There are aphidious species in the braconid group that will only target your aphids. So they go in, they inject their eggs, and they don't need to mate to inject eggs. They can lay eggs that will become male. They only need to mate if they want a female egg. So they can spread like wildfire over an entire area. So where something like a ladybird would only take out a handful each day, a single wasp could take out some 50 aphids in the space of a couple of hours with ease because of how efficient they are at injecting and reproducing. So they kill hosts very quickly, and it's used across the world. There are multiple countries, including here in Ireland, that have companies that sell the ideal parasitoid for your pest species. In Africa, there was the issue with the cassava plant. It was getting decimated by the mealybug that had become invasive there. So they went and got its natural parasitoid from South America, bred it in captivity, and then basically let it loose on the wild invasive bug. And within a decade, the cassava plant was saved. The crops were saved. So it made a huge difference to have a natural control over an invasive species. And it worked. So they are very important from an economic perspective, but that also means they're important for the overall health of an ecosystem. These guys keep insects that are very good at reproducing, that are very good at just taking over areas, keeping them in check. It's a very important point in that. How to tell if you already have parasitoid wasps. So this is the kind of thing for yourself. If you have a back garden, you likely already have them. But if you want to confirm and report, oh, I've got this particular species, I didn't know it was here, you're going to have difficulty because without a microscope and without any kind of experience or familiarity, you're not going to be able to tell what you're looking at. These guys are very, very small, and even under a microscope, which is what this picture is from, you still are nowhere near enough close enough to tell certain distinguishing parts. Sometimes you need to literally count the number of little pits on the head, number of antennae, wing venation, how many little veins make up the wing. These are all things we use to determine what the wasp is, and that's very difficult when they only are about a centimeter in length or smaller. So for yourselves,
to identify them and find them, really what I can recommend is really just seeing what you see. If you see an unusual wasp that doesn't look like it's your typical social wasp, odds are it is a parasitoid. So they're usually slender. They look more like they belong in a as in like the movie with the aliens and like the xenomorphs, because they essentially are. They are the xenomorphs of the real world. So if they look a little bit almost like alien in appearance, then you kind of have an idea it's a parasitoid. So if they will come into your house. They are nocturnal. They will look for other insects because that's what they're looking for, hosts. And they can be very tiny. This little calcid wasp was on my finger while I was actually writing up notes for this presentation. So they can just appear out of nowhere, funnily enough, right in front of you. So if you open up your eyes looking for them, you will see them. So some of the things to kind of discern it's a wasp is this pinched abdomen. So between where the thorax ends and the belly abdomen begins, there's this little pinched thing, and that's where so that's a common trait among wasps. And then obviously the wide-eyed head as well. So if they're on hosts though, that's where this kind of image is kind of showing you what you're looking for. You're looking for the cocoon. So with aphids, they just mummify them. They just go creamy colored or golden colored, and the wasp just emerges. But on caterpillars, there's usually cocoons. So they eat the host inside out, and then they emerge, wrap themselves in the cocoon, and then become adults. So if you have caterpillars and they're destroying your garden, if you happen to see one that's covered in all these cocoons, wait a couple of days and all the other caterpillars are going to get attacked as well. So it's a very effective way of keeping pests because a single caterpillar can produce dozens of adult wasps as a result. So how to attract them to your own space? How do you get them into your garden, which I imagine is where most people want to get the benefit from these guys. So they are pollinators, so having flower beds and similar things, whatever you're doing to attract bees and stuff like that, the same thing applies for parasitoid wasps. They are inclined towards daisies, so plants with dill, fennel, alyssum, cilantro. These are plants that will encourage parasitoid wasps to stick around because it's a good food source. Shallow pool of water. All insects need some kind of water supply, even just a small amount will help. Even bringing some hedges and shrubs, really just kind of allowing more areas for shelter where insects will happily migrate to and nest near and maybe lay their eggs there. That'll again attract the wasps that want to inject their babies into those eggs. And same for your general garden, just for the grass and everything, leaving patches of uncut grass just a little bit longer than you might have normally. So just letting your kind of garden get a bit wilder, let a bit more wilderness come back into your garden. And if you're like me again with a back garden and you've kind of decided to do a bit of home farming with some carrots, strawberries or what have you, if you have them, you're going to invite parasitoids with you as well, because they will follow the food supply. They will go for some distance following the signals of their host. So if you've got aphid problems or if you've got caterpillar problems, then the wasps will be there. So if you create a scenario where you're attracting the pest, you're going to attract the parasitoid. And since there are wasps that actually target the parasitoids, so yes, the parasitoid will target the parasitoids, you're also attracting a whole ecosystem then, a whole hierarchy of food chain. So the end point for this really is that if you build it, they will come. If you allow your garden or your wildfire patch, if you want to do something with tidy towns or anything like that, it's just letting a more natural environment come to the area. Let different flowers bloom, let different stages come along, and and the whole idea is, like, if your plants are being destroyed by insects, it's not an end of the world. It's part of the natural cycle. And if you have pests, if you have insects that are going to town on your veggies, then that will in turn attract the wasps. They will come to where the host is. And if you are wanting to hedge your bets, there are places that do sell them to farmers because of that. And even large places, if you ever happen to have a bottle of alcoholic cider, you may not notice it, but there's probably a little parasitoid wasp swirling around in it, because they are using fruits that attract aphids, and as a result, wasps sometimes get picked up in the process. So it's more about realizing that these guys are already here. They're already playing a huge role, more than what we know, because there's so many of them. So it's more about letting your garden play its part. Let the wasps come in and do what they're supposed to do. So if you let your garden be a little bit more wild, you'll have the parasitoids coming in droves. So thanks very much, guys. Hope you enjoyed the talk. Hope you found it interesting. I'm happy to answer any questions you might have about anything. Obviously, like that, this was kind of scratching the surface of what's involved in parasitoids in general. So yeah, thanks very much.
Okay, thank you, Conor, of the recording of Conor. Are there any questions? Yep, Peter.
Hey, cheers. Thank you very much, Conor. That was really interesting. One of the cool things is that our lab has previously looked at parasitoid wasps as well as an alternative to chemical pesticides, and if you apply entomopathogenic nematodes, it doesn't harm the wasps, which is what we were afraid of, because if the wasps were attacking infected cadavers, could the IJs travel out and attack them? But they don't, thankfully. They work synergistically together. But I was just curious about what you were saying about the fact that the females, obviously they don't have the XY chromosome, the females can asexually reproduce essentially to have the male offspring in it. But if they mate, they can produce a female. But I was just wondering, are they like bees where if they mate, they've basically got inseminated eggs forever? As in, can they store it? Do they have a spermatheca, or do they have to mate relatively often?
Thanks, Peter. Yeah, that's actually how they control it. So the females can control how many eggs they make female male. So they have they have a little section in the ovipositor to store the male semen, and basically they can choose to eject it if they find a more appealing male. So they have complete decision over who's their baby daddy, basically.
That's really cool.
Yeah, it is really interesting, but there's other species. There's a Vetrapennis species that actually lays a single egg if it needs to reproduce, but when it lays the egg, it results in a 3,000 offspring when they emerge from the host. So it just multiplies from a single egg, which is madness. But it's why there's a book by Donald Quick. He covers like the whole general area of parasitic wasps. And it's like the number of different internal biochemical things that goes on with these guys. It's head spinning, which is why it's so fascinating, because like even like they've been studying for so long, but there's still a huge gap of information. They actually made Charles Darwin's been quoted as saying that these guys made him doubt the existence of a kind omnipotent God. So they've had a huge impact on that. But yeah, they're fascinating in terms of reproduction as well. It's interesting to know. That's kind of why I asked the question about you about whether other insects, whether or not they could coexist with the nematodes. But it's interesting to know they did.
Yeah, they work synergistically together, so they're better together if they're applied together on a field rather than either on their own. Cool. What book was that? Donald Quick, did you say?
Yeah, Donald J. Quick, Quicke, 1997. It's just called Parasitic Wasps. Okay. He's a retired researcher now, but he did another book in 2016 as well that was more detail orientated at specific parasitoid groups, and it's just a wealth of knowledge. I think most university libraries would have at least one copy of one or the other. If you want to buy them, it's a head spinning amount of money. So yeah, library's your best search bet.
Okay, thank you.
Yeah, thank Norris.
Okay, Karzan, is your hand active again, or is it still lingering? Okay, I guess it's still okay. Perfect. Okay, if there's no other questions, then we move on to the final talk of the evening, which is by Karzan. Karzan, do you want to quickly introduce yourself?
Thanks, Inga. Actually, I have introduced myself in the presentation, but I am introducing myself again. So I am Karzan D Ahmed. I am a PhD student in the Applied Ecology unit here at NUI Galway. So I have been working with insects in general, and my interest in particular is Diptera flies. So my today's presentation will be about an important group of insects rather than bees. So it will be hoverflies, Diptera sylphidae. And I'll be talking, it will be actually just an introductory presentation about hoverflies and how you can recognize them, and then how you can record your selfie records to the national selfie database, the Irish national selfie database. So I hope you enjoy it.
Okay, thank you, Karzan. Let's see if I got it shared. Click, click, and
hi everybody, my name is Karzan D Ahmed. I'm an entomologist and a PhD student in the Applied Ecology unit here at NUI Galway. Today I will be talking about a very important group of invertebrates, which are known as hoverflies, Diptera selfie. So my presentation aims to focus on the following points. First of all, an introduction to hoverflies and their ecosystem services, recognizing hoverflies, differences between hoverflies, bees, or wasps, identifying hoverflies in the field and by photographs, submitting hoverfly records to the National Selfie Database, and some simple guides about hoverfly identification. So currently there are more than 930 species of Selfidae known across Europe, and within that 185 of them are recorded in the state here in Ireland. They have 4 developmental stages, as we can see here. For example, for the adults start lay eggs and then they hatch into larvae. They have different larval stages based on the different species of hoverfly, and then they go into pupa stage, then after that into adults. Okay, so there are important ecosystem services provided by hoverflies. First of all, in the adult stage, hoverflies are known as important crop pollinators, and they play a significant role in global crop pollination. For example, yields of more than 75% of globally important crop species are enhanced by animal pollinators in general, and of course, hoverflies playing an important role in that. But also beyond yields, animal pollinated crops provide important micronutrients and minerals for human diet, for example, vitamin A, the carotenoids, and also fluorides. In addition to crop pollination, so hoverfly larvae many hoverfly larvae play an important role in the biological control of agricultural pests such as aphids, as we can see in this picture, a larval hoverfly feeding on an aphid. So there are some general characters for recognizing hoverflies. First of all, hoverflies are usually shiny or reflective, but that's not applicable for all hoverfly species. And many species of hoverflies are black with yellow, or sometimes they have other pale markings on the body. Hoverflies, they have also veins parallel to the trailing edge of the wing. They are called what we call a false wing margin, as we can see here. So that's a false wing margin for hoverflies that can be used to recognize hoverflies. And also hoverflies have vena spuria, which is only available in hoverflies, but that's also sometimes could be difficult to see in the field, as we can see here. So we sometimes need a microscope to check vena spuria, as we can see here in a hoverfly species, Platycalus angustatus. Then it's also important to recognize and differentiate between male and female hoverflies, because sometimes you may get confused between male and females. And in terms of recording, it's really important to differentiate between male and females. Here on the left side, for example, we have a hoverflies, and if the eyes are meeting, so we can be sure that a hoverfly is a male hoverfly. As we can see in self-observation male, that could be used as a character to recognize males. But here on the right side, so we have self-observation female, and if the eyes don't meet, so we can be pretty sure that it's a female, but that does not mean it's always female, because some species, they have also males where the eyes are separate. In other words, if you have a sylphid fly, the eyes are meeting, so we can be sure that it's a male. But if the eyes are not meeting, it could be male or it could be a female. But in most cases, it will be a female hoverfly. Now, it's also important to differentiate between hoverfly versus bee or wasps in the field, because sometimes we may get confused between the three insect groups. For example, here we have a hoverfly, Sericomia silentis, a honeybee, Apis mellifera, and a social wasp, Vespula sp. So for hoverflies on the left, we can differentiate them by depending on the eye characteristics. So the eyes are large for hoverflies, covering most of the head, and they usually have shorter antennae with only 3 segments. But on the other hand, for bees and wasps, the eyes on the sides of the head are not big as in hoverflies, and they not cover all the head, and they usually have longer antennae with 12 or 13 segments. Hoverflies have only 1 pair of wings, but for bees and wasps, they usually have more than 1 pair of wings, so they have 2 pairs of wings, but that can be very hard to see on live insects. For hoverflies, they are usually fast hovering flight for most of the species, but for bees and wasps, they are slow flight and not hovering except in very few species. And hoverflies, they don't have a pollen basket, but for female bees, they usually have a pollen basket. So that's another character we sometimes may use for differentiating between hoverflies and bees, or in particular bees. And we have some hoverflies also that can mimic bumblebees, and sometimes they cannot be recognized simply by sight in the field. So we need to know some important features to differentiate between hoverfly and bumblebee. And these specific features are really important to ensure a correct identification. For example, for hoverfly, we have short antennae, but for bees, they usually have longer antennae, as we said, more than 12 segments for most of the times. And hoverflies, they have large eyes, and bumblebees, they usually have small eyes. It doesn't cover the entire head, but as it does in hoverflies. And hoverflies have only 1 pair of wings, but bumblebees, they usually have 2 pairs of wings. Okay, many hoverfly species can be identified directly in the field with some experience. However, it's really important to look at some critical features. For example, leg color, shape, wing venation are required. In this picture, for example, Tropidacita, a hoverfly species, it's really important to look at the hind legs and get pictures of the hind legs. That triangular shape confirms its identification. In addition, open structured flowers such as hogweed in sheltered sunny locations are usually most productive locations for getting good views of hoverflies, as in this hoverfly species, Helophilus pendulus, but for many other species as well. Okay, sometimes hoverfly can be identified in the field by photographers. However, photographs need to show critical features such as typical photos from above often do not show important features such as leg color and face pattern. For example, here for a thick-legged hoverfly, Psitta pipiens, it's really important to take photographs of hind leg or from the side so that we can see the critical features for the identification of that species. Here on the right side, we have the same species. We can identify them as a male hoverfly in general, but we cannot identify into species because the legs are not visible. So we cannot be sure that it's a thick-legged hoverfly. So that's why it's important sometimes to get, for the same species, to get more than 1 picture so that you can get an accurate identification for a particular hoverfly species. Okay, so there's a national sylphid database for Ireland, and it's held by the National Biodiversity Data Centre, and it's available to view through biodiversity maps in this link. So also you can submit your self-read records online, but all the records will be validated before being added to the national database to get an accurate identification. I'll show you in the next slide. Here, for example, for submitting a hoverfly record, first of all, you need sample details. For example, recorder name, recorder email address, and recording date, county, location, and also grid reference or spatial reference of the sample. And then next, you need observation details. For example, species name, species picture, life stage, whether it's an adult or larva, and then sex, whether it's a male or female, and then abundance in terms of numbers, and survey method. In fact, I didn't cover survey method in this presentation because of the limited time, but of course, there are many methods for collecting hoverflies in the field. And here you need a determiner name. Sometimes it could be yourself, or sometimes you need your, for example, you send your sample and it's identified by an expert, so you can add the person who determined the hoverfly species. And here sometimes we need scientific keys for some species that could not be identified based on pictures. So we can mention the identification literature or the key that we used for identification of that sample, of that particular sample. And here you can also add foraging on, for example, the name of the plant that you recorded a hoverfly, and here any additional information for a particular species. For more information, you can click on that link and you will get that into this link, and more information is available here. Finally, there are some recommended simple hoverfly identification guides. Here on the left side, Beginner's Guide to Hoverflies in Ireland by National Biodiversity Data Center. And this is available for free in this link. I have put it below. So you can download it and it will be very useful for beginners. The next one is Britain's Hoverflies by Stuart Ball and Roger Morris. Is a very good identification guide as well, because it contains many color guide and illustrations for almost all the hoverfly species recorded here. And however, this is not available for free. So if you want to use it, you may need to buy the book in hard copy. The final one is by this link or website by Stephen Falk, and this is a very good website for hoverfly identification based on photographs. So if you have your hoverfly photographs of a particular species, you can compare the features with pictures that is presented here in this website, and it will be sometimes very useful to get a good identification of your hoverfly samples. Thank you very much, and for any questions, please email me at k.dahmed1@nuigalway.ie.
Okay, thank you, Karzan. Let me just exit that. Okay, great. That's our final presentation. So thanks very much. Does anybody have any questions for Karzan? Okay, no, it's coming up. So I have a question. If you're looking at bees and wasps and let's say hoverflies as pollinators, do you know what percentage of like ecosystem services in terms of pollination is from hoverflies as opposed to bees, let's say?
Well, actually, bees are the first most important pollinators in general, but hoverflies, in terms of classification, so they have been Diptera actually in general, they have been named as the second most important pollinators in terms of crop pollination. Okay. And in general, the value of pollination actually is about $200 to $500 billion. And that's based on annual, according to a study. And of that, like in terms of crop pollination, 35% of global food production crops are dependent on crop pollination. Of course, hoverfly play an important role in that. And would hoverflies have different preferences, let's say, over certain plants? Bees or? Yeah, there have been some studies actually, like in terms of plants, but in general, like in terms of flowers, so they mostly like yellow and white flowers because they are open, and in general, like safely, they have short proboscis, so they can easily access those flowers to get food from them. Other flowers they may visit, but they cannot access like pollen easily. Like for example, like bell-shaped, like the pink flowers. It's not like very attractive to hoverflies, although they may visit. But in general, like yellow and white flowers are the preferred flowers for hoverflies.
So if you wanted to attract them to your garden, that's the ones you would plant.
Exactly.
Okay.
Yeah.
And that would also be where they lay their eggs and where the larvae hatch, or do they usually lay their eggs in different locations?
It depends on the species, Inga. For example, for some species, they probably lay eggs in like inside like crevices, dead holes of trees. For if they are pollinated, for example, if they are biocontrolled, so they lay eggs in proximity to, for example, field crops in grasses, so where aphids are found, so that the larvae hatches and they attack the aphids. So it's pretty much depends on the species.
Okay.
Yeah.
Okay, great. Thank you. Any other questions for Karzan or any of the other presenters? Okay, if not, then I think we're already a little bit over the scheduled time. So thank you very much again for everybody presenting and for everybody joining in to listen to it. The recording will be made available somehow online. I will let you all know. I can send everybody who signed up the link for the recording, and then email as well where the presentation will be put up eventually. Okay, so everybody have a nice evening, and
thanks very much for organizing this.
Yep, no problem.
Thanks for organizing this, Inga.
Okay, great. I'll
Bye.
see some of you soon.
Thanks, Inga.
Bye.
Thanks. Bye.